The overall goal of this single filter-based workflow is to easily and quantitatively purify N-glycans and proteins in tandem from biological samples for mass spectrometry analysis.
The scalability, speed, and analytical reproducibility of this method makes it directly accessible in regards to resources and skill sets to biological mass spectrometry laboratories. The main advantage of this technique is that it allows researchers to investigate glycomics, glycosylation site occupancy, and proteomics questions simultaneously in large-scale, high-throughput biological studies.
We've overcome the challenges of N-glycan measurements by coupling this method to a mass spectrometry-tailored derivatization strategy, resulting in accurate relative quantification between cancer and control states.
With reduced analytical variability, new biological interactions may be elucidated between the glycome and proteome, yielding key insights into disease states and new opportunities for biomarker discovery.
To begin, load 2.5 microliters of plasma onto a filter. Then add 2 microliters of 1 molar dithiothreatol solution to each sample in the filter. Dilute the sample with 200 microliters of 100 millimolar ammonium bicarbonate PNGase digest buffer. Cap the filter sample tube and lightly vortex, being careful not to disturb the filter from its seat. Incubate the sample at 56 degrees Celsius for 30 minutes to denature the proteins and glycoproteins. To alkylate the samples, add 50 microliters of 1 molar iodoacetamide to give a final concentration of approximately 200 millimolar. Then, incubate at 37 degrees Celsius for